Thank you, Matt, and hello, everybody. So, nice thing about being Professor Emeritus, you get free parking. So, I'm going to talk to you about some work that Jerome Goudet and I have recently submitted to genetics. So, Jerome from Switzerland has been here several times, and he asked me to pass on his greetings to those of you who he met during his visits. You probably don't know Stephen Hazlitt. Steve's a statistician in New Zealand, and it's one of the bonuses of me moving back is I've got to meet and then collaborate with Steve, who's publishes quite frequently in the linear model space. So he is teaching and trying to rescue Jerome and I from our theoretical lacks. So those are my colleagues. We're talking about the work that Jerome and I have done, and sort of getting ready to talk about the work that Steve was working with us on. So a series of recommendations talking about if we have to estimate, I'll call them descent measures, things like inbreeding and co-ancestry and FST and so forth. If we're going to estimate them, there's some things we should keep in mind. And I've just written them here, and then I'll come back to them as we get to them. They're not in order of importance, though, in order of appearance in the slides. Okay, so we have a linear mixed model. We have a series of observations, some individuals, some measured trait maybe, and we posit that these depend on some fixed effects, age, sex, and so forth, and maybe some particular genetic variants, some particular SNPs that we'd like to see how they affect the trait. And then there's an overall random effect, the overall genetic profile of the individual. And this is the information there, among other things, contains information about the relatedness and the dependencies among the individuals. And then there's an error term. So the sort of classic, don't make a long way now, quantitative genetic models let us write down the variance of the observations in two parts, and there's a lot of assumptions behind this, but it's simplistic. There's a genetic component involving what we call the genetic relatedness matrix, and a components of variance called here the additive genetic variance. All the alleles in the genetic profile are assumed here to act additively. So it's probably not true, but it's a good way to get started. And then an error term. Sometimes this G is written as a K isn't used instead of G, so G is 2K. So we have observations, we have some fixed, a coefficient matrix. We would like to estimate the fixed effects and make statements about the variance components, the genetic and the non-genetic variance components. So that's where we're going. To do that, it's convenient and helpful, historical, to work in the language of measures of identity by descent. So really talking about the dependencies amongst alleles in the profile. Initially, civil rights work back in the 20s talked about correlations. And more recently, since for a while now, from America, we're talking about identity by descent. So two alleles are identical by descent if we can trace them back in time to some convenient reference time, and these two current alleles are copies of the same ancestral allele. And then we typically say that this reference population, otherwise not very well defined, but otherwise consists of alleles which are not identical by descent. So everything is relative to this reference population, at least initially. So that's the historical background. And then we've got this notion, because we're talking about things that happen over time, we recognize that what we have now lets us predict what will happen in the subsequent generations genetically. On the average, we can't predict exactly what will happen. You know, my wife and I have two children, same process, different outcomes. So when we talk about the probability of identity by descent, we're really talking about the expectation of identity state over all the possible replicates of that evolutionary process. So when we sample a population and observe the alleles, we have a frequency in the population we sampled, typically call that P, but that is itself one realization of the allele presence and the allele probability I'll write as pi. So what is this genetic relationship matrix? And the reason I introduced these IBD measures, so theta is a probability of identity by descent, and this is the element in row J, column J prime. Two individuals in the sample, their relatedness is described by 2 theta, where theta is the probability of identity by descent, a random allele from individual J, a random from individual J prime, if they have this common ancestral allele identical by descent. If J and J prime are different off the diagonal, then we call the theta, we call that the co-ancestry coefficient of the individuals. If on the diagonal, the diagonal elements are 1 plus the inbreeding coefficient over 2 of the individuals. So those are parameters of interest. Classically, quantitative genetics goes back primarily in the plant and animal breeding field, where populations could be manipulated. They were experimental populations largely. So it was possible to calculate, to predict these IBD measures from the pedigree. So these were almost always pedigrees were known. The thetas could be calculated by simple rules, as I'll show you in a minute. But the pedigree might not be known. And even if the plant and animal breeders thought they knew it, they might not have written it down correctly. So there might be some errors in the pedigree. Moreover, the pedigree, the expected values of the identity status are different from the actual values. And it's the actual values for the individuals we're studying that really matter. It's helpful to know what we think they have in terms of relatedness and so forth. We'd like to know really what they actually have. And in general, we understand that we cannot estimate these parameters of interest, these thetas, largely because we don't have the reference population, and in particular, the allele probabilities are not known to us. There are some exceptions, of course, but we're going to concentrate on what happens if we estimate something else, some estimable function, instead of the parameters of interest. Is that going to upset things? And one of the things we've realized, belatedly maybe, the parameters these thetas look to be the ones of interest, but they may not be the relevant parameters. And we're thinking that these estimable quantities are indeed the ones that are important. So prediction is easy. Here's an individual, J, and his 2 parents, J prime and J double prime. And at some point in the past, there's a pedigree, this ancestor they have in common. So what's the chance that J gets identical alleles, meaning coming from the ancestor, or that the 2 parents receive identical alleles? And every time we go through one of these individuals through a meiotic process, there's a 50% chance either of those alleles gets transmitted. So we take a half to the power of the number of individuals in this pathway. And then maybe the ancestor itself had identical alleles, that we put that in if that's the case, and we add over all the possible ancestors and all the possible pathways. So it's a fairly straightforward calculation. We can calculate the probabilities of identity by descent if we have a pedigree. Go ahead.
Sorry, I'm just not sure how you're defining theta. Is it within the variation in theta? Is that variation within a given pedigree, or variation over the population process that generates the pedigree? Within pedigree variation, obviously, in the meiosis, and we also have the between pedigree.
I'm thinking of the between pedigree. So even though I've got the pedigree written down,
okay, but you're thinking of it as a particular realization of the pedigree of the
yes, yeah, absolutely, yes. Thank you. Yes. So these probabilities are expected values over all the actual things, which allele was actually transmitted when we come to each parent. One of those 2s is, and on the average, it's, we expect 50% chance.
Yeah, but there's still within pedigree and between pedigree variation.
Okay, I'm not following the within pedigree. All right, thank you.
This
Thanks.
is something I've been thinking about.
Okay, good. All right. I'd like to know that. Good. Thank you. All right. So what's the problem? If we have the pedigree, we're fine. We can predict these values, and then we've got a GRM, and then we can do a lot of stuff. We don't have the pedigree very often, or it might not be accurate. And how can we use the genetic information, either from the current study or some other source, that we've got information that might lead us to sensible values that we can use in place of the inbreeding and co-ancestry coefficients? So there's only really a one equation. Alleles or IBD, if they have a common ancestral allele in some reference population. If theta is, this is any two alleles for the moment. If theta is the probability that they're identical, what's the chance that they are both a particular type of allele? We can observe the allele type. A is a generic type. What's the probability that this pair of alleles are both the same type? They would look the same to us with our typing technology. Theta of the time they're identical, so there's only one copy independent between them. Pi is the probability. 1 minus theta, they're not identical, so we've got two independent copies of the allele, pi squared. So this is a probability, but it refers to an observable event, the allele state. So that's something that we can observe, the allele state, which suggests that we might be able to estimate the parameters from the observed values of the allele states. And we're going to suggest we can't do it quite that simply, but there's certainly been a long history where good progress has been made. So if we focus now on two alleles in one individual X, what's the probability that individual is homozygous for that allele? Now the theta, the generic IBD parameter, is the inbreeding. So if of the time the alleles are IBD, we've got one copy, or they're not IBD, we've got two copies. So now let's set up a little indicator, zeros and ones. Let's say that we're going to score the individual's observed genotype as heterozygous, score that a 1, or homozygous, score that a 0. So this individual would be classified, would be scored as a 0. So that's an observable quantity. It's either 0 or 1. The expected value over all replicates of the history of that individual depend on the unknown allele probability and the inbreeding coefficient. So now H tilde, this is an observable quantity. So let's put in the observed value instead of the expectation. And we should be able to solve. Certainly for F, we just simply divide what we see, H, by something that we will use instead of the pi's. And so back in the 1950s, the observed allele proportions in a sample were used in place of the pi's. And here's a very simple classical estimator of the inbreeding coefficient for one individual. And of course, this is generally just averaged over multiple genetic markers. So that's nice. And it's often written as 1 minus the observed over the expected heterozygosity. We can certainly see whether the individual's heterozygous or not. It's not clear that we should be using heterozygosity for this, this function 2p 1 minus p. And in fact, the Masayoshi and they call it gene diversity. Maybe more sensible. Now, the problem with doing that is that the expected value of that denominator over evolutionary replicates is not just what we're looking for, what we were hoping it was. It depends on the co-ancestry of all the pairs of individuals in the sample that gave the p tilde and the inbreeding coefficient of all the individuals in that sample. And I'm sort of embarrassed to point out that this square brackets term is, we gave it a name, Clark, Cockram and I, in 1969. It's not a widely cited paper, but anyway, there it is. It's the group co-ancestry coefficient. For large sample sizes, we don't have to worry about ends of sample size. Let's forget about that. But we do have this confounding fact that what we're taken as an estimate is modified by the average co-ancestry of the sample individuals. So that's a problem. And then I just point out that we're going to have multiple SNPs these days, as my colleagues know. We've got lots and lots of SNPs. We could average those ratios, or much better, maybe take the ratio of averages. Much better in the sense we're not going to be worried by very small denominators, get large estimates. Sometime colleague, John Story, had a nice application of the central limit theorem and said that, in fact, these statistics are such that asymptotically this ratio expectation is the ratio. Expectation of the ratio is the ratio of expectations for a large number of SNPs. So that was comforting. Now, Elizabeth had a classical paper in 1975. It's good. She was at that time thinking about non-inbred individuals, and two individuals in a pair could be classified according to their genotypes. And basically, they were scored as 0, 1, or 2. They had two pairs of alleles with the same, two A's, or one pair, an A, and the double heterozygotes had two A's and two B's. So Elizabeth had a nice likelihood approach and would estimate the probabilities of 0, 1, or 2 pairs of alleles in identical by descent for two such individuals. And this method is quite widely used. It's in the Plink package. So we can observe allele state. We can see what our typing technology gives us as an indicator. And I'm not going into mutation processes. So I'm just supposing, just for the discussion, is that the alleles we see, if they have remained without change since their ancestral allele, they're identical by descent and they'll be the same. What we see is their allele state, that's identity in state. So we would like to estimate the IBD probability so we can get that genetic relationship matrix so we can do all our quantitative genetic work. And we can't do it directly. And I keep quoting Elizabeth again. She says identity by descent is relative. It's not an absolute quantity. So I like to think about this hypothetical case where a community is set up, composed, all the founders are first cousins to each other. It's a large community, and it's a great, it might be a family reunion. They all get on well, and they say, let's do our thing as a family, first cousins. It's legal, so they pair off at random, and they have children. And so what do we think is going to happen? Well, if I'm one of those family members and I collect some saliva samples and do some typing, nothing's happening. Because all the parents, all the founders were equivalent. They were all within that community. There's no pair different from another. They're all, I know they're cousins, but they don't appear different, any pair from another. And their children are fine. They're not inbred. So everything is good, except Matt's sitting over there said, those are weird people. They're all cousins and their children are inbred. So I'm right, Matt's right, with different perspectives. So it matters. So our references, it matters. There's no such thing as relatedness as an absolute quantity. I talk about first cousins. I know what first cousins are. I don't know what it really means genetically unless I know about the population that their parents belong to. So we often forget that. I don't often forget it anymore, but we used to. So for a long time now, there's been a series of estimators for these IBD probabilities. So this was the one. The language was in this GCTA package. It goes up, I think, even further, and it's widely used in the animal breeding context. So each individual has a series of genetic markers, SNPs, and can be scored as 0, 1, or 2, how many copies of a particular allele the individual carries. So we've got some discrete data, and those are the dosages, X, and if we average the dosages over all the sample individuals, every homozygote gives us a 2, every heterozygote gives us a 1. So if we add up the X's, we're getting the total number of the reference allele in the data, and we divide by sample size, we get twice the sample allele frequency. So we have a dosage with a sensible average. And then it's common to think, well, maybe these sounds like count data, we're counting the alleles. Why don't we treat the X's as binomial variables and think about a plug-in estimator for their variance, just 2p, 1 minus p. So let's use that to standardize the dosages. And then to sort of carry that logic forward, take the dosage for one individual, subtract the sample mean, second individual, subtract the sample mean, and divide by the product of their standard deviations. So that's a nice little ratio, and that was put forward in the Yang et al. As an estimator for IBD, the current system. They took the average of ratios. It's much, much better to take the ratio of averages, because this term can be very small and doesn't behave well. And in this format, Story and Ochoa showed that we've got asymptotic unbiased, asymptotic in the sense of large numbers of SNPs. So the problem, of course, is that the one I've just alluded to with the inbreeding estimate, although the sample allele frequency is a good estimate, it's unbiased for the allele probability. When we square the estimate, we don't get an estimate of the squared probability. And of course, the difference becomes this average current history. It turns out, if we little bit of algebra, that what that standard estimator is actually estimating is what the authors claimed they were estimating, the current history, but it's relative to the average current history each individual has. So they've got their current history, but each of them separately is related to members of the sample. And then there's an overall relatedness measure for all the pairs of individuals. So they're actually estimating this quantity. So we would like, if we can't estimate theta, we'd at least, I think, like to estimate, this is my second recommendation, would like to estimate quantities which at least had the same rank as the unknown. We would like not to be too bothered by our sample. If I add an individual to the sample, I don't want to change my estimate ranking for the relatedness. So
theta j with one subscript is the average.
The theta j is the average. Yes, it's the average. It's that individual's average current history. So that's pretty. And we didn't make this up. We noticed it. Story noticed it, and other people have done too. So let's not use that method. It's become to be called the standard method, and it's widely used. So let's do it slightly differently, and let's talk what we've called allele sharing. So we're going to take the individuals and look at their SNP profiles and ask how many alleles do they share. It's reminiscent of the early work on the numbers of pairs of alleles IBD. So we've got two people, three possible genotypes, and we're going to score those pairs as a 1 if they have the same homozygote, a 0 if they're different homozygotes. Otherwise, it's a half. Just they've got two people, 4 pairs of alleles, 1 from each, and the proportion of those pairs which are the same allele. And then we'll probably average this over all the SNPs in our data. So that's our, I'm going to call it A, allele sharing. Maybe it's not obvious why I call it A, but anyway, this is the sample allele sharing statistic for 2 individuals. For one individual, the allele sharing is 1 for homozygotes and 0 for heterozygotes. So the allele sharing for an individual is just a complement of the heterozygosity. For two individuals, it's sort of like the diversity thing that came up in the inbreeding estimate. So those are our statistics, and we're going to form estimators for two individuals. I'm going to switch my words from co-ancestry, the IBD probability, to kinship, K. So the kinship of two individuals is estimated as their allele sharing relative to the allele sharing for all the pairs of individuals in the sample. And the expected value of this over replicates of their history is the parameter of interest relative to the average co-ancestry. So this, these estimates in expectation have the same ranking as the target parameters. And the actual numerical values depends on the sample, the kinship in the sample. By construction, for different individuals, these kinship estimators sum to 0 for one sample. But in inbreeding, take the individual, their allele sharing 0 or 1 compared to the sharing between pairs of individuals, and that parameter is estimating the within population inbreeding coefficient, the actual inbreeding compared to the average co-ancestry. So we've preserved the ranking. So the logic is sort of embarrassingly naive and simple. We have a question. We have a set of alleles of interest, and we set up a statistic, which is the degree of allele sharing for that set of alleles, typically 2 alleles per individual or alleles from 2 individuals, but it can be more extensive. We've got an allele sharing statistic and an expectation that depends on the unknown allele probabilities and a target parameter, a target theta. We don't know either, and we don't think we can estimate theta unless maybe we knew the pi, and we're not sure we want to say that. When we form those ratio estimators in expectation, the top and bottom lines involve the pi's, 2 pi, 1 minus pi. But it's the same in the top and bottom line, and so that the pi's cancel out. We don't need to know. That's recommendation 3. And just going back to Ochoa and Story again, these estimators are good in the sense that they're unbiased, not going to make great claims for variance, but they perform quite well. So Ochoa and Story come out with the same logic at the same time we did. And John and I presented our work at NC State University in 2016, the same meeting, and said, look, we've got doing the same thing. Why don't we publish jointly, or not jointly, but try and get them published back to back. So the editor of genetics agreed, and we each submitted, and ours was accepted, and John's wasn't. Well, to be fair, John had 2 papers, and the editor wanted them combined. And John said, no, I've got so much stuff, I've got 2 papers. So he didn't, so his work didn't come out till 21. But what they were doing, he and his colleague, instead of comparing the sharing to the average sharing, they compared it to the minimum. So it's a trivial difference. We've got a slight preference for means as opposed to minima, just for stability of the estimates. So what they're estimating is the target parameter compared to the minimum value. So later they submitted their work to PLOS Genetics, and I got to review it 3 times. And eventually I got them to agree to write in the paper, Weir and Goudet did not estimate IBD probabilities. So that was, it seems like a trivial thing, but it was a major concession. But then they said, but actually we want to estimate IBD probabilities, and our ratios can be negative. Some pairs of individuals are less related than others. They don't like that, so they want to be positive. And then just a couple of years later, Ochoa had another paper said the Weir good A estimates have a uniform downward bias. But our method in their software package is the only unbiased estimator under arbitrary relators. That's a very strong and interesting statement. It looks like a fallacy. How can they claim to be estimating an IBD probability when they don't have these allele probabilities? They don't have the reference population. So there's a very simple solution why they can, how they can do that. They set the minimum value to 0. So if the minimum value is 0, then they are indeed estimating this target. So that's a little, I think I call it putting the rabbit in the hat, but they have a justification. They call that an oracle estimate. Right, let's look at some data. The 1,000 genomes is a nice data set. A lot of SNP data, 26 populations around the world, some Native American in red, the yellow are African origin, got the Europeans, the Southern Asians, and the Eastern Asians. So we can estimate with allele sharing, inbreeding, and kinship. So all those 2,000 individuals, 2,200 individuals, on the x-axis we've plotted the allele sharing inbreeding estimate. On the y-axis, the kinship estimated for each individual. And this is all relative to the whole world, the whole data set, just on a common reference. We've got the Africans down here, the reds, the Native Americans, we've got the Europeans in blue, purple, South Asian, and green, East Asian. Now, these are not probability estimates. I've written them as an inbreeding coefficient, as inbreeding relative to average co-ancestry. You can't see the scale. This is 0.05, pretty big numbers for inbreeding. But what strikes immediately is that this line's not horizontal, nor is it vertical. There's a general positive correlation. So it's very difficult. And then I've just taken away the complicated African populations, which are different, and the highly admixed American, and just looked at the Asian and European. There's overall positive correlation, but it's certainly not a simple one. So that's our recommendation. If you want to estimate inbreeding, don't forget about co-ancestry and vice versa. If you want to estimate co-ancestry, probably shouldn't forget inbreeding. Looking at the inbreeding coefficient, this is the allele sharing, colored Africa, Native America, Asia, Europe, Asia, and this is each individual's allele sharing compared to the average allele sharing for pairs of individuals in their own population. And over the whole world, things don't vary all that much around 0. There's a little departure from 0, plus or minus, but no continental group stands out apart from these highly admixed Americans. Some of these values are very high, and that's been commented. These are values which are on face value or even higher than we would expect from first degree parentage, full sibling or parent-child parentage. So now if we change our reference from their own population to the world, one African person sharing versus the sharing for pairs of people, any pair of people in the whole study. Now Africa's clearly different, clearly much less inbred. And we know that it's a very old population. It's been a long time for mutation to increase genetic diversity, a lot of diversity. And the rest of the world has markedly higher inbreeding coefficients. The same thing for kinship. Here's the kinship. There's a much smaller picture. The kinship for the local population is reference, the world is reference. So those are the allele sharing estimates. Here are the standard estimates. And we've got not much going on locally, but now on a global perspective, Africa looks to be the most inbred or the most related, which sort of doesn't jive with our understanding of human evolution. And if we look a little closer, look at the standard estimators, compare on the X and Y axis, do we use the local population or the world population? Africa and the Americas, things don't behave particularly well. So the reference matters. It matters less for the European and Asian. Now, the Brownings, Brian's here, they have some nice work on using quite a different approach where they say, well, we can't observe identity by descent, we can't observe the history, but we can look for a region of the genome which for an individual, say, is completely homozygous, a long region. So that's probably the only sensible description or explanation of that long region of homozygotes is that the two copies of the genome the individual gets, one from each parent, they are identical by descent. So we're looking for long runs of homozygosity, ROH. We've got a direct observation inferred to be of identity by descent. So the fraction of the genome in such regions, because there's a direct estimate of the inbreeding coefficient. And this is shown on the y-axis and the allele sharing estimate on the x-axis. And indeed, they seem to have, they're quite congruent. They've certainly got the same ranks, a little bit of a departure from 45 degrees when we've got the Africa and America. So these are, I mean, they're good estimators. They're not estimators of IBD, however, because they're conditional on the determination of a region is long enough. So this, I mean, it's based on experience, obviously. So it's a very good surrogate. They're good estimators, and a little bit more work to get them, but I think we're going to get the same answer as by allele sharing. If we use the standard estimators, it's just a real mishmash, so we don't use those. There's been other approaches. There's a lot of buzz around this King and King robust methods, where those authors said, let's estimate relatedness for a pair of individuals if they're not inbred. And moreover, let's just use those two individuals, the target individuals, without worrying about the complications of population structure, all that stuff. So their estimate is very simple. And Matt showed, I guess, was in your thesis, it was certainly about that time, showed that what it was actually estimating was the target parameter, but it was affected by the inbreeding of those two individuals. The authors liked their method because it said we're just using the individuals of interest. We don't have to worry about the others, which seems to be ignoring the whole problem. If relatedness is relative, why would you, anyway, why would you do that? And why would you do it numerically? Well, you're going to get this very strange answer. The allele sharing estimates, which we know are ranked the same as the true IBD probabilities on the x-axis, the King estimators on the x, y, and the y-axis, and they're all over the place. So why would you ever use them? These are real data. If we filter the data to get rid of inbreeding to some extent, things look better, but still seems to be no reason to use those estimates. Everything I've just said, I could change the word individual to population. So we've got this notion going back to the civil right of F statistics. So here are our take on F statistics. We say, let's look at a series of populations, the average allele sharing for individuals in each population, averaged over populations, compared to the average sharing for pairs of individuals in a population, averaged over pairs of individuals, over pairs of populations. So this is the within population, quotes, inbreeding coefficient in what civil right called FIS, individual compared to subpopulation. Or we could compare individuals sharing to the average sharing for individuals from pairs of populations. So we've got individual compared to distinct individuals here, distinct pairs. Here we've got individual compared to distinct populations, FIT, and FST is the variation among populations, sharing within populations compared to sharing between populations. So we've tried to make the notation as close as we could to right. Wright's in our symbols, his statistics, were allele sharing within versus allele sharing for random pairs of alleles. So we said, let's do one individual. Let's look at their two alleles compared to the alleles from different individuals. Is there something special about being in an individual? Wright said, let's look at these two alleles and just compare it to a random pair, which is fine, except that the random pair might be from the same individual. And for the whole population, a random pair of alleles from the whole population could be from the same population. We've got a population effect and or the same individual. So we've got a very, very small difference most of the time for large sample sizes. This AW is the same as our AES for large samples. The AT, the one that's and this is also Masatoshi Nei's notation. The AT is for random pairs of alleles from anywhere. We generally do not have a lot of populations. We don't have a large number. So this is the one that's going to cause Wright's estimators to be different from ours. Masatoshi Nei followed Wright very closely, and I am really embarrassed, and I'm going to do something about it, and I'm doing it now, partly. Cochrane and I had a long, vigorous debate in person, in letters, and in print, saying, silly old Nei, he's got all that wrong, and Nei saying, silly old us, we've got it all wrong. We didn't mention in 1986, Nei changed his definitions, and his are exactly the same as ours. So Nei's estimators are the allele sharing estimators. So that's nice. I wish Nei was still with us, but anyway, we'll certainly put that in print. There's been a lot of discussion about, we have other critics. They said, why don't we refer to Hudson's work? Well, Hudson certainly has done nice work, and he and his colleague had a paper where they collapsed the data from genotypes to alleles and then estimated FST from allelic data. I mean, that's fine. You can, and maybe you only have allelic data in some cases. What you've done, however, is to confound your estimates by the difference between inbreeding and current history. But you could confound by Hardy-Weinberg disequilibrium. If there is Hardy-Weinberg equilibrium, their estimators are identical to our allele sharing. Going back even further, we have this best-selling paper, and what we did was to say the populations are independent, which is clearly not the case. So what we were actually estimating with the Weir and Cochrane estimators is any particular theta, relative to the theta between. And Cochrane had pointed that out in 1973. This is what we were doing. So if you think that this ratio is biased estimated because it's not estimating theta, you're correct, because it's got theta b. For equal sample sizes, the Weir and Cochrane estimators are exactly the allele sharing ones. So now we can get started on what I want to talk about a little bit, but we won't take long. So let's put this in context. How can we use this? So one quantity of interest is the heritability. It's a statistic. It's the ratio of, we look at the variance of a trait as the genetic variant component of the variance divided by the total variance. So it's not a biological thing, but it's a very convenient statistic. So to estimate heritability, we need to estimate the genetic and the non-genetic variance components. And it's a lot of work. I mean, this is good. A lot of people done a lot of nice stuff. I'm going to just follow a simpler case by Balding and company, and where they take for the we've got the variance, we've got a genetic variance, we've got a variance of a trait value. We've only got one individual, so we can take the sample variance over a set of individuals. let's take the sample variance of the trait values for n individuals and get the sample variance and use that as the estimator of the variance of the trait variance. And then if we do some regression or do something nice, we got some residuals, and then we can get the estimate of the residual variance, and we can construct the regression variance. We construct an estimate of the heritability, the total variance on the bottom line, and the genetic component on the top line. So I'm not sure how general this equation is, but it's very nice. Going back to our quantitative genetic model, where we said that the variance of a trait is the G times sigma squared A plus sigma squared I, sigma squared E. The variance of the trait can be written in terms of the trace of the GRM and the sum of the off-diagonal elements. So that's and for our purposes, that's a very convenient formulation. But now, if we look at what our GRM is in terms of identity coefficients, we can see that what we're estimating depends on the inbreeding and the co-ancestry history of the sample we've got on the top and the bottom lines. If there was no inbreeding and no co-ancestry, individuals were all independent, the estimated heritability by that method is indeed the definition: additive genetic variance over total variance. However, this is the expectation. Now, classically, the classic literature for heritability says that when you've got inbreeding, that affects the heritability, and by a term, a factor 1 plus the inbreeding coefficient, where this F is the IBD probability. So that goes back to Douglas Falconer's book and so forth. We don't have a G, we don't have the thetas, and what we do have is these kinship estimates, the Ks. So we've changed the question. We're not estimating G, but we're going to plug something in. We're going to plug in, instead of the G, we're going to plug in, for example, an allele sharing kinship matrix, where the elements of that matrix are their allele sharing compared to the average between individual sharing. And the expected values of the elements of this matrix, 1 plus the within population inbreeding on the diagonal, and the kinship. Oh, I've got to drop the subscript. I don't know what that is. Shouldn't be there. It's the kinship estimate. So that's what we're doing. I wonder if I can keep using this nice equation, and I'm not sure. Taking the trace and the off diagonals for our kinship matrix. If I do that, however, it turns out, going my way through, it turns out that what I am actually estimating is 1 plus the within population inbreeding, which is a very nice result, and it looks like it should be correct, but I haven't got the details quite squared away. Now, the surprise maybe is if we use the standard GRM, grind our way through, we get the same answer. For a large sample, there's a little correction, sample size too. So though the two matrices are very different, the elements of the matrix, when we collapse those matrices down to two numbers, the trace and the sum of the off diagonals, put them together to estimate heritability, we get the same estimate algebraically, and we certainly see that numerically. That's one application. Inbreeding depression is another one of interest. This is a very bad reproduction of a publication. This is human height, where the authors had a lot of people whose parents were related, typically first cousins or more remote, occasionally, unfortunately, closer than first cousin. So we've plotted their observed height for these samples of people against increasing inbreeding coefficient. And there's a pretty clear linear trend as predicted. So the trait mean is predicted to change linearly with the inbreeding coefficient, and it does certainly empirically.
So those are the data estimated inputs.
The ROH estimate. Yes. The actual, the actual.
And the vertical lines are the powers of two.
Those are the known relatives, degrees of relatives, I think. Yeah. And I couldn't get a better, but I found. So, and just the text, subjects with kinship as though they're offspring a full sibling or parent-child, which we don't like that, but it certainly happens. Those are significantly shorter than other people, shorter than those whose parents were uncle, niece, or half siblings, and shorter than those who were first cousin, offspring of first cousins. So their analysis was sound. They included fixed effects, age and sex, and some population structure things. So Jerome's student Lavanche had a paper last year, just putting things in a linear models, mixed linear model context, using one of the fixed effects was now the inbreeding. The beta was the effect of inbreeding on height, and so the coefficient had the inbreeding coefficient, had the X matrix had the inbreeding coefficient, and then we got the genome Y, the whole profile for their genetic profile. So it's straightforward linear algebra, except you've got to be careful which package you use, because these matrices, these kinship matrices and so forth, are not positive definite. So some packages don't like that. When it comes to matrix inversion, other packages manage. So that's what we've been using. And she did some numerical work. This is not a very easy slide, but so the simulations were such that the regression coefficient of height on inbreeding was the slope was negative. Height goes down with inbreeding. The scaled regression coefficient was -3, which is the black line. So these blue estimates were using the allele sharing GRM. I think the first ones are the pedigree. Pedigree values. I'm not going to read easily. The allele. This is just using linear models, and then using the mixed linear models. Things behave pretty well whether we're using the actual pedigree inbreeding coefficients, which we knew for the simulation, or the allele sharing, or the standard, or the ROH estimators. And we do better with larger sample sizes. So things seem to be hanging together quite well. And then finally, I think we can do association mapping. We can look for the SNPs that affect the trait, used 1,000 genome of individuals and to construct trait, gave them trait values. And just say here's using and then the allele sharing matrix, genetic relationship matrix, refers to the relationship of the individuals regardless of the source, regardless of whether it's close familial relationship or deep evolutionary relationship. So we don't need principal components. So the red dots, there are 5 causal SNPs, so they pop up at the top of the line. Those are the most significant. The very most significant was not causal. So, over here is the standard GRM with some principal components. And here we've got a couple of plots using the method that at that time was used in our own center, using this combination of PC air and PC relate, where there's a relatedness matrix and there's principal components. And we do better, of course, with the principal component for population structure in that context. But I think we do better still without doing either. And then finally, I just mentioned in passing this recent Ochoa paper, where they were set, their conclusion was that the association results are robust to the GRM. Doesn't matter which GRM we use. The title of the paper said they were robust to common kinship estimation biases. I think bias is an unfortunate choice of words. Anyway, so I won't say any more about the paper. We're thinking about it now, but here we are. So what have we done? Kept your rapt attention, thank you. So we've got IBD probabilities that we'd like to use, like to estimate them. We don't quite manage to do that, but we've got some estimators which seem to work pretty well, but a lot of context. And more to come. So thanks for listening.
So you, somewhere in the middle, you said, I won't say anything about variance. Your estimates are all inconsistent,
Yes.
that's not really biased, you're sharing about and so on. And it's nice, it's lovely stuff. But in a small population,
Yes.
or with a smaller number of SNPs,
The number
how
of
much
SNPs,
you lose
the number of
by
SNPs,
not worrying about allele frequencies at all.
the number of SNPs is critical. We've gone down. With using 1,000 genomes, we've gone down to samples of the size of 1,000, using 1,000 SNPs, which is about as small.
1,000 SNPs with
yeah.
decent allele frequencies, or
Yes.
1,000. That must also
Just randomly. So we don't like to filter on allele frequency, because that certainly changes the answer. Changes the question.
Yeah.
Changes the question. So by reducing the number of SNPs down to 1,000, it seems to work pretty well. We'd much rather use it. The sample size, the number of individuals, has very little effect in general. But that we knew that, I think. Sample size.
Sample size on which you're doing the thing.
The number of
But
individuals.
your allele frequencies come from a larger sample.
That's right.
So you've got a small population of an endangered species, and that's all you have.
That's right.
The
So
allele frequencies are these.
yeah. So we would
That's critical, in other cases misleading.
we would suggest that see what happens if you don't use allele frequencies. Use the allele sharing methods. And so the expected values do not depend on the sample size.
Right.
So,
Right. Yeah, that's nice.
yeah. Go ahead.
Follow up on, I have a sort of similar question.
I'm not. I should have repeated. I should have repeated Elizabeth's question. Sorry.
Yeah, right.
So what about sample sizes, number of individuals, number of SNPs, number of individuals less and probably not very important, number of SNPs critical, and 1,000 seems to be good. So
So I'm going to follow up. It was the number of SNPs. I was asked more about the number of SNPs. That's sort of two related questions. One is just the number and spacing. I mean, think about some
Yes.
space so you can avoid disequilibrium. But I was wondering if you look at things like where you get the data, say, from sequencing, as opposed to from a SNP array, which has all sorts of weird biases in terms
Yeah.
of how those SNPs were discovered.
yeah.
Are there differences, or have you not? looked
Yeah.
at
So
that?
what's the effect on the sampling of the SNPs?
That's only,
How do you choose them?
identification.
I'm going to say something heretical. It doesn't matter.
Well, that's what.
It doesn't matter. I don't care if they're next to each other. I don't care if you've got adjacent SNPs in your mixture. It doesn't matter, because we're asking the question, has this individual got more sharing than pairs of individuals with this set of SNPs? Some of them are banging next to each other. Some are on different chromosomes. It doesn't matter. So there's no point in filtering on LD. All we're doing is comparing an individual with the average or a pair of individuals with the average. And we do this. So in that context, it almost doesn't matter what we do.
But what do I infer about the biology? That's a different question.
I don't think if you want to put an evolutionary model on, you can say, well, what I see is a result of migration or selection. I could put it in a model and then try and predict the values and then compare my estimates to the predictions. But I don't think there's no measure of relatedness, not an absolute. I think all we're trying to do is in our quantitative stuff, all we're trying to do is to rank individuals. We've got good ranks. We know which are the most inbred, we know which are the most related, and are the most related most likely to share genetic profiles.
So if you have good ranks, is there a way of using the ranks? say, these are the ones I'm really interested in.
That's what, in fact, we're doing.
Okay.
We are using ranks, yes. The nice, yes, pretty good. I can't tell you how long it took me to think that through. We kept getting questions. Well, what about LD? Why don't you filter? What about allele frequencies? All that's interesting stuff, but I don't think we need to do it. Nice. Anybody online, Matt? So I've sent the slides to Deb. No, so it was, yeah, I think Matt has them. And if anybody wants the manuscript, be glad to share it. We haven't got a review yet. It's not going to be an easy process, I think. But anyway, we'll see. So thanks, everyone.